Good morning everyone and welcome to the session led by Working Group 3 on field and lab methods. We've got some great talks lined up on the programme today which are going to really showcase the depth and range of work that's been taking place over the last few years. But we wanted to start with a bit of an overview of what we've achieved as a working group. There have been two main streams to Working Group 3. The first is building capacity for DNA-based monitoring through knowledge sharing and training. And the second is building consensus through workshops, experiments and ring tests. So I'm going to give you a brief overview of what we've achieved both in terms of building capacity and consensus, starting with capacity. One of the most effective tools we've had at our disposal has been the short-term scientific missions, or STSMs, funding knowledge exchange and collaboration visits between institutions in different countries. In Working Group 3, 22 STSMs have been completed, with knowledge flowing to and from 21 different countries. These have covered a wide range of different species groups and protocol elements and have led to numerous research papers and ongoing collaborations. We've also run training courses with a real focus on enabling participation by scientists in ITC countries, which tend to have less research resource. This was a training course run in Bucharest in October 2018, which saw 20 participants from 12 countries gain experience in sampling and analysis of eDNA. Meanwhile, there's been a really active diatom group which ran 4 training schools between 2018 and 2020, each hosting around 25 participants and again covering both field and lab methods. Now, the plan had been to really increase the focus on training courses and capacity building in the final year of the DNAqua-Net programme, but unfortunately, like so many other things, this was thwarted by COVID and we weren't able to run as many as we had hoped. But we have been able to do a lot of work on consensus building. Now, taking leadership on this working group, I have to say, was pretty daunting for us. There were a couple of hundred scientists signed up from the beginning, and we did an early survey when the working group first started, which pretty much confirmed our fears. There were an even greater number of protocol variations. So our challenge was how to make progress in building consensus without just spending 4 years arguing with each other. So we started by throwing out the idea that there's one ultimate protocol to rule them all, and approached it instead with the recognition that there are almost certainly several different perfectly valid ways of doing each step of every analysis. But that doesn't mean the answer is to give up on consensus building and standardization, because this is crucial as the methods move out of the research world and into widespread application. So what's happening now is that we're moving from a situation where one knowledgeable researcher or research group has full oversight of the whole sampling and analysis process, including interpretation of results, to a situation where you might have a contractor collect the samples, a commercial lab analyse them, someone else might do the bioinformatics and data analysis, and then ultimately the end user has to use the data to inform decision making. And sometimes those decisions are really expensive. So we need to put in place systems and frameworks to enable the decision makers to trust the results and to interpret them correctly. And that's what we're talking about when it comes to standardization. And when we framed it like that, we found there was much more consensus than it originally appeared. So we set out trying to answer the questions that had been asked by the end users. They're basically faced with this really quite daunting array of methods in the scientific literature. And this can be really off-putting and lead to the conclusion that the technology just isn't ready for use in the real world yet, when actually we know it can already provide so much valuable information that can't be obtained using conventional methods. Our consensus building activities have generated 4 broad types of outputs. We've made tools to help guide interpretation of results. We've taken the first steps to creating formal European standards that set out the core principles of certain steps in the analysis process. We've undertaken methods comparisons and ring tests to start to understand how consistent the outputs can be when samples are processed in different labs and with different methodological choices. And we've produced best practice guidelines in various formats. So we'll start off with the methods comparisons and ring tests. There are 2 ring test projects underway. One for diatoms focusing on DNA extraction and PCR steps, which we'll hear more about in a talk later on today. And one on benthic macroinvertebrates, focusing on the same steps and also incorporating a comparison of different DNA capture methods. Both of these ring tests arose from DNAqua-Net workshops, the microinvertebrate one at the University of Porto in Portugal and the diatom one in Limassol, Cyprus. Now, the point of a ring test is to see whether different labs can get the same results when they apply the same predefined protocol. For fish, we did something a bit different. And here we showed that even when you have different choices being made at almost every step, you can still obtain really consistent results, both in terms of species composition and relative read counts. And this paper is being written up at the moment. So moving on to the tools we've built, the most notable is the validation scale. This was the product of a workshop in Innsbruck in 2018, and it sets out a framework for determining the extent to which a targeted single species assay has been validated, and for interpreting results obtained from assays at each level on that scale. I'm not going to go into any more details about this here because Bettina will present it in one of the talks later on in this session. We've also started working on formal standardization of DNA-based methods through SEN, the European standards body, and I'm sure this will be covered in much more detail in the Working Group 5 sessions. So the diatom group have already incorporated DNA-based methods into the SEN technical report on benthic diatom sampling from rivers and lakes. And a draft standard on aquatic eDNA capture has been submitted to SEN as a new work item proposal. This was the result of a really fruitful collaborative exercise started in a workshop in Sarajevo in 2017 and continued in Pech in 2018. For me personally, this has been one of the most interesting exercises because when we all sat down initially, the starting point was that almost everyone did almost everything differently. So it seemed like a really impossible task to embark upon. And most of us, whether we're in academia or in commercial environments, spend most of our time trying to differentiate ourselves from one another. But as soon as we turned the conversation around to ask, what do all of our different methods have in common, we discovered a really strong common core that we could start building the standard around. And we had exactly the same experience at a workshop in Rome in 2019, when we started the same process for sediment sampling. So we had one day of highlighting how we all do completely different things, and a second focusing on the fact that most of the basic principles were the same. So this hasn't made its way to a standard draft yet, but there's a manuscript in preparation that sets out recommendations that could develop into a new standard draft. So finally, the best practice guidelines. And these are aimed at practical applications of DNA-based monitoring. This guide to eDNA applications was published last year by Jan Pawlowski and Florian Altamat. It was aimed at biomonitoring in Switzerland, but of course has relevance far beyond that. And it's got really lovely illustrations and step-by-step instructions, and is generally a great read if you're into this kind of thing. And meanwhile, a larger group has been working on another set of guidelines with a slightly different focus. In the Practical Guide to DNA-Based Methods, which will be published as a PenSoft ebook, we synthesize our collective knowledge and experience and set it in the context of the practical and logistical constraints faced by environmental managers. We highlight the key decisions and trade-offs throughout the process and consider what might influence the decisions and how they in turn influence either the end results or other factors downstream in the workflow. And we also describe the quality control steps needed to provide assurance that results can be trusted and acted upon. So the idea is that this will be a useful tool both for people who are new to the field and choosing a methodological approach to use, and also for end users commissioning DNA-based monitoring work who will have to evaluate proposals that may vary quite substantially in the proposed methods. So to summarise, we've seen a really big shift over the last 4 years, and DNA-based methods are being rapidly adopted across Europe and beyond, even though it'll take some time to integrate into the WFD. And we've actually got a pretty good level of consensus around the main principles of field and lab methods, even if the details vary. Having said that, there's no one silver bullet protocol. There are loads of inherent trade-offs and still a lot of areas where further research will bring improvements. So our challenge is to standardize methods so they gain the maturity that they need for widespread adoption, while also encouraging continued development and innovation. So I want to finish by just saying a huge thank you to everyone who's contributed their time, expertise, support and good humour to Working Group 3 over the last few years. I know a lot of people have put in a huge amount of effort, often in their spare time, often late at night, to make a success of the workshops, the training schools, the meetings, and to produce the resources we've created. I think we've made a significant collective contribution to the growing up of this field, and I've certainly had a lot of fun along the way, and I've loved making new friends all over Europe. Thank you for listening and enjoy the rest of the presentations.